This is Ken Beers with Oxford Life Sciences. In this presentation, I'll be discussing the economics of late-stage drug development in progressive diseases. Progressive diseases span a variety of conditions, from rare genetic disorders with poor survival to common conditions where health only slowly declines over decades, but they pose common challenges to developing drugs. One is that the drugs may slow but not reverse disease, limiting measures of clinical value and health technology assessments. Another is that progression timescales may vary across patients or across stages of disease, which would impair the ability of a clinical trial to detect a therapeutic benefit without stratification or things like prognostic control. These then pose several significant design questions when you're entering a late stage of development. Should I pursue all patients or only early stage patients who have more opportunity to benefit? Should I narrow down to just fast progressors or those in late stage disease where I can detect a treatment effect more readily? How many patients do I need to study and for how long in order to have a statistically significant treatment effect? And how much beyond that should I go to be credited with clinical value by payers and physicians? All of these questions then end up impacting the sales of an approved drug and risk-adjusted program value. So here we're going to use integrated clinical and commercial simulation to explore these questions and use this simulation as a tool to design and optimize programs. The basic context for what we're going to consider is the following. We're heading into phase 3 for a drug that we've just completed a successful phase 2 program that demonstrated proof of concept with clear, strong efficacy data. We assume going ahead in phase 3 that we're going to be using the same intervention protocol and endpoints and dosing as we did in phase 2, or at least one of the arms of phase 2. But we have the opportunity to change inclusion criteria, change the size of the number of patients, and the duration of time over which we observe them. In addition to phase 2 data, we also have registry data, which contains natural histories of patients over finite periods of time, potentially over longer time periods than we may observe in a relatively short phase 2 trial. The approach that we're going to take is from these two sources, registry data and phase 2 data, we're going to learn a model of disease progression over time, both for untreated patients and for treated patients. We're going to then set the design of our program, inclusion criteria, sizing of the trials, duration of the observation, and we're going to use these patient models to generate efficacy outcomes for the trials according to the statistical plan. We're going to then apply a cost structure and safety risk assumptions to get the program outcomes where for each stage, trial, or regulatory review, we would see how long it takes, how much it costs, and what's the chance of success. We then take all those outputs from these clinical program simulations and link them to a commercial forecast and valuation model to predict overall value of the late-stage program. We're going to demonstrate this approach for 4 generic disease prototypes that vary in prevalence and severity of disease, using for each one very approximate, rough kind of assumptions just to get the flavor of each type of disease. Obviously, within each one of these categories, in real life, there would be very significant variation in all the different assumptions we're going to make. We have 4 diseases, starting first with ultra-orphan, which has 1,000 patients in the US and progresses over a time scale of 3 to 5 years. Orphan, 20,000 patients in the US with a time scale of 5 to 10 years. Specialty, 200,000 patients progressing over 10 to 15 years. And primary, with 1 million patients in the US progressing over 20 to 25 years. For each one of the diseases, in addition to the US, we're going to model EU, assuming the same prevalence rate, and so the disease population in the EU would be scale proportional to the US based on general population. Now we need to create this synthetic data that we're going to have, which is going to be patient registry data, and it's going to be the phase 2 data. And to do that, we need to use underlying models of the disease dynamics that in practice we won't know. We're only using these to generate the data, and then we're going to have to learn from the data what these models are separately and use those in our calculations. There's 2 components of the disease dynamics that we have. One is the time scale of the progression of disease, which is going to vary from patient to patient according to probability distribution. And the second is the shape of the change in quality of life as the disease progresses from time of onset equals 0 to the complete progression at the end of the time scale. For the timescale distribution, we see the values for each one of the 4 diseases at bottom left. In each disease, we assume that there's a 50-50 mix of fast and slow progressor subgroups, and that within each one of those subgroups, there's additional variation. And so we get a wide variation in how fast and rapid patients then go through this common disease process. At right, we see the mean change in quality of life as a function of time of the extent of the disease, which is going to be equal to the time since onset divided by the progression time scale. We have 2 different shapes that we consider. One is linear disease, in which quality of life declines relatively uniformly with time. And the second is nonlinear disease, in which in the early phase, when quality of life is above 80%, we have a very slow decay, starts to accelerate in mid-stage disease, and then rapidly accelerates in end-stage disease. As we'll see, even these 2 diseases, if they have the same population, they have the same timescale distribution, yield very different commercial forecasts and outcomes under health technology assessments solely due to this shape of distribution. As I said, we don't know the underlying dynamics, and so we use those actual models on the previous page merely to generate patient registry data, and then all we see is that registry data, and we use that to try to infer, again, the underlying dynamics. At bottom, we see for ultra-orphan linear and nonlinear disease, example synthetic registry data here for 50 patients each, being followed up to 10 years at random time since onset or until death. What we do is we take that registry data and we take the general form of the disease model that we just outlined, and we have to relearn the shape of progression and the distribution of timescales across patients in order to match the registry data. What we see below is the blue shows the actual curves and distributions, the orange shows the fitted values of the data, and we see that we do a pretty good job of fitting the disease progression module to the registry data, and we see similar performance then for the other diseases that we consider. To generate synthetic phase 2 data, we also then need to model the treatment effect of our drug, which we assume slows but does not reverse the progression of disease. And to understand how this works, we consider the figure at bottom left, where at an initial time since onset T0, we have a patient who has a baseline quality of life, and then we either make the decision to treat the patient, in which case we set A equals 1, or we do not treat the patient, we have a control, A equals 0. If we have an untreated patient, they continue to progress just as they did in the natural disease evolution in the patient registry. If they're treated, effectively the treatment slows down the rate of progression according to a treatment effect tau, which again is going to be something that varies from patient to patient. So essentially by treating it, we have a patient that undergoes the same evolution of dysfunction over time. It's just everything happens more slowly. At bottom right, we have the treatment effect distribution we use in each disease. In primary, in red, we have a relatively low treatment effect. As we go to smaller and smaller disease populations, particularly orphan and ultra-orphan, we have higher treatment effects because often in those diseases, we're pursuing drugs that target the genetic drivers. Given the underlying actual disease dynamics and the treatment effect, we can synthesize a phase 2 data, which again, in practice, is what we're going to be seeing, and then we have to infer the treatment effect from that phase 2 data. For phase 2, we use relatively small trials with larger patient counts as we go up to more prevalent diseases. When we have ultra-orphan and orphan diseases, we use synthetic controls, so we only have a single arm. Especially in primary, we have randomized dual-arm trials. And then for each one of these diseases, we both have a look-back period before baseline, where we establish a prior medical history to get some idea about a rough estimate of the progression timescale and the time since onset of that patient. And then we observe the patient for protocol duration from 1 to 3 years, growing as we have more slowly evolving diseases in primary. At bottom left, we see one example of synthetic phase 2 data for the nonlinear primary disease, with the controls in blue, untreated, and the treated patients in red. From that data, we infer in the plot at bottom right the fitted treatment effect distribution, and we compare that to the actual distribution we used in blue to generate the data, and we see that it's reasonably close. Generally, we end up overestimating the standard deviation or the variance of this distribution because of individual measurement errors, but we do a pretty good job of fitting the mean treatment effect, and as we go to bigger and bigger phase 2 trials, we do a better and better job of fitting it. So as we go into phase 3, we assume that we have some registry data and we have this phase 2 trial data, so we have at least approximate fitted models of the treated and non-treated progression of the disease over time at different times since onsets, considering all the variation that we see in terms of progression timescales across different patients. We now can use those models to simulate individual patient response in clinical trials that we define and design based on a set of inclusion criteria, a size of the trial, so the number of patients in each arm that we use, and the protocol duration, the amount of time that we track each one of the patients after baseline. We're going to consider 3 sets of inclusion criteria, with early mid being the most common. Here we're taking all patients in an early or mid-stage disease, which means quality of life above 20%. Once you get to the late-stage disease, there's really not that much benefit to be gained from initiating treatment, and so the broadest that we use is this early-mid condition. Now, in trials when we have an early-mid patients, particularly in nonlinear disease, we have a lot of different variation over the individual decline in quality of life that we would see over any 1, 2, 3 year period. And so in order to increase the power of the trial, we consider 2 alternative, more restrictive inclusion criteria. One, where we only include patients in the mid-stage disease, which in nonlinear disease is where you start to see a more rapid rate of decline. And finally, the early-mid patients, but only those that we estimate from their prior medical history to be fast progressors. So if their estimated progression timescale is less than the median across all patients, we would include them into the trial. The only reason we use these latter 2 inclusion criteria is just to improve the power of the trial, particularly in nonlinear disease. The problem, as we see, is that we may end up restricting the breadth of the label, and that has a negative economic impact later. Once patients pass the inclusion criteria and are admitted to the trial, they're randomized to an arm, either A = 1 for treated, A = 0 for non-treated controls. And then we use our fitted models in order to generate final quality of life values at the end of the observation window, at the end of the protocol duration. And we do that based on the treatment effect that we generate for a patient if they're treated. What we're assuming here is that the phase 2 treatment effect holds up in the pivotal studies, but what we could do is introduce a random mechanism of action or dosing risk by randomly setting the treatment effect to 0 if we wanted to introduce more uncertainty into our late-stage outcomes. The primary endpoint that we use is a change in quality of life from baseline. So a higher value of Y means that we retain more final quality of life and have clinical superiority. We model a 5% annual dropout rate, and for patients that drop out, assume pessimistically that their final quality of life is equal to 0, in line with oftentimes what we might see from regulators, and then apply a one-sided statistical test, non-parametric test, to test for efficacy. We use the patient models and the trial simulations to determine success based on efficacy, but we need to combine that as well with success on safety, and we do that by generating, with 10% probability, a random adverse safety signal that, if it occurs, means that the trial fails. We set that 10% by using published studies that look at why drugs failed in late stage and attributing a certain fraction of those to safety as opposed to efficacy, commercial, or some other unknown reason. In this study, 17% of late-stage failures were due to safety, and that suggests overall 10% of late-stage programs would fail due to safety. Of course, that varies considerably by therapeutic area, by whether it's a novel drug or not, orphan, etc. We're taking here a relatively broad value for progressive diseases outside of oncology. One thing to point out, and it's a potential problem if you're solely using analogs in order to get the probability of technical and regulatory success, is that in the study they found that 22% of those failures were attributed to commercial reasons. But of course, the entire reason why we're doing this economic analysis is to see if the program makes sense economically. And so we don't want to apply a haircut for failure on commercial reasons up front, because we're going to make that decision later. We have the potential to double count. This approach that we take here then avoids that potential double counting. We model development from phase 3 onwards, but we're going to vary the regulatory pathway according to the disease. In ultra-orphan and orphan disease, we assume that we can gain an initial approval just with a single phase 3 study, but then we have to conduct a separate phase 4 confirmatory study, and only if that's successful then do we gain full approval. If it fails, we're pulled from the market. In specialty and primary, we have to take the traditional route where we do 2 parallel phase 3 trials and have both succeed then in order to be able to apply for full approval. In ultra-orphan, because there's so few patients, we use synthetic controls for those trials. And here, when we have the phase 3, phase 4, for simplicity, we're assuming we use the same trial designs for both phases. We could go back in very easily and modify the approach if we wanted to use a different set of assumptions for phase 4 when we're confirming a treatment effect than we do in phase 3. For each one of the individual trials, instead of using an analog to estimate the probability of technical success on efficacy, again, we're using a trial simulation. So we want to recruit patients, assign them to arms, generate synthetic outcomes, apply the statistical plan, and see did we meet the statistically significant cutoff that we needed in order for that trial to be a success. For recruitment, we assume that we're going to be drawing patients across a network of academic medical centers, and in the smaller diseases, we assume that these academic medical centers are both fewer in number and collectively treat a greater fraction of the prevalent population. And this is what we see oftentimes in rare diseases, where you might have relatively small numbers of centers of excellence, where patients go not all the time for routine medical care, but maybe once or twice a year for a checkup and to get their treatment plan modified. And so we have a greater leverage of a relatively smaller number of centers than in specialty and primary, where many different hospitals and major medical centers treat these patients, and we also have patients being treated outside of an academic medical center basis as well. Now for our recruitment, we're going to look at how the size of the trial and the number of patients that we want to draw into our trial affects the amount of time that it takes for recruitment to proceed. And to do that, we use the methodology that's discussed here, where we draw an initial pool of in-network patients, assuming 50% are willing to participate in the trial. And for each one, we generate, according to the prevalent population, a random time since onset, progression time scale, treatment effect, and time to the next HCP visit. Then each month, where the recruitment is ongoing, patients who are due for a visit come in, see their HCP, and are screened based on inclusion criteria. Those that are passing and willing to be admitted are randomized to an arm and admitted if the quota for that arm is not yet filled. And if they're admitted, they're depleted from the remaining pool of candidates. And then each month, those patients age and they progress, and we have newly incident patients arriving, and we continue this process drawing and recruiting at random until the arm quotas for each arm are filled. Now what we see in the figures at bottom right is for the top linear ultra-orphan and bottom nonlinear primary, what recruitment looks like when we use a network of 10 academic medical centers. On the figures at left, we see how many candidates are remaining within the network when recruitment ends. And at the right, we see the trial duration in years, which is equal to the recruitment time, plus we assume 6 months to start up the trial before recruitment can begin. We include the protocol duration, and we include 6 months of analysis after all the data is collected. In addition, when we later compute costs, we assume that there's a year of follow-up for each patient in an extension study that adds to the cost but doesn't delay the time that it takes in order to report top-line results from the trial. For linear ultra-orphan, what we see is once we move beyond about 75 patients in the trial, we exhaust all the candidates that are in network, and it takes a long time, years, for new patients to slowly trickle in order to be able to fill quotas that are larger than 75 patients. So if we need to do a larger trial, we need to use more than 10 sites, but of course there's only 100 sites that are out there. So we see immediately that these ultra-orphan studies are very difficult to recruit. For nonlinear primary, we have an abundance of patients, and so even with very large trials, we only deplete the candidate pool within these 10 academic medical centers only modestly, and so we can complete these trials and complete the recruitment quickly. We, for primary, look at 3 different inclusion criteria: early mid, but just the fast progressors, and just mid-stage, and we see, as we would expect, that the more restrictive inclusion criteria we use, the fewer candidates that are available, the faster it is to deplete them, although here we're nowhere close, and the more times it takes to do recruiting, because we just have to wait that little bit longer for the smaller number of patients to come in. Once we've simulated recruiting for a trial, we know how many sites we have, we know how long the sites need to remain open to recruit patients and then to follow them, and that gives us the information we need to estimate what the overall cost to execute a trial is. We assume for simplicity a constant cost structure across each one of these diseases that includes central costs, a cost incurred per site, and a cost incurred per patient that are both fixed costs and monthly variable costs. The only thing that changes as we move from one disease to the next is the cost of goods sold, or the production cost of the drug that it takes to treat a patient every month. That's going to be higher for these very rare ultra-orphan diseases, since oftentimes we're talking about a biologic, versus primary disease where we might have an oral that's much cheaper. So again, we're only using here very approximate, rough estimates. Real trials are going to vary dramatically in terms of the assumptions that we use, but just for illustrative reasons, we look at what would these calculations tell us about the cost of a very small trial versus a larger one. In the small trial, we have a single arm, just 75 patients. We only follow patients for 12 months, and we have a relatively expensive, per patient COGS of $1,500, assuming that it's a more expensive biologic. And when we calculate the cost, we find that this would cost about $9 million to do this trial. By comparison, in a larger disease like primary, where we have 2 arms, 500 patients per arm, follow patients for 36 months, but the cost per month to treat a patient is relatively low, these large number of patients and longer duration means that now we have a significantly more expensive trial at about $51 million. From the trial outcomes and individual patients, seeing how the change in quality of life evolves from baseline to the end of the trial, we can calculate cost effectiveness metrics like quality adjusted life years. The figure at left shows the observed QALYs that we would obtain over a trial of protocol duration specified, where what we have to do is we have to wait, starting from the same baseline in both arms, to see these 2 progression curves diverge over time. And unless we wait for a very long period of time, we really don't observe that much difference between the 2 curves, and the actual observed QALYs are quite low. In order to make these drugs look anywhere close to being economical under a cost effectiveness framework, we have to make an assumption that the treatment effect persists beyond the observation period. And so we assume if we start patients in treatment at the same time since onset in the trial, they have the same kind of mix of the progression timescales patients in the trial, and we keep treating the patient beyond the window observed in the trial and see the same slowing in the disease over time, we calculate what this extended QALY benefit is. We then calculate from that benefit a target annual price by valuing each QALY at 40,000 and assuming we avoid an additional 100,000 in medical costs per QALY obtained, and we divide that value then by the duration of the treatment needed to realize that benefit. We assume by default in the EU that they apply a cost effectiveness framework somewhat like this to our drug, and so if we have a list price higher than that, even if we can command that in the US, we assume that if we're going to get reimbursement in the EU, we have to discount down to that target annual price. We also use these QALY benefits in order to compute what the peak preference share is, assuming that the bigger the benefit that we show in a trial, the more physicians are going to use the drug in practice. And so for each one of the diseases, first based on a level of competition, assuming roughly in ultra-orphan and orphan, there's not much competition, and in specialty and primary, we have one head-to-head competitor. We have a peak preference share that's going to vary between a minimum and a maximum value, and we use the QALY value to interpolate between those two, so that if we have a very poor QALY benefit, we're going to tend to be at the minimum, and a very strong QALY benefit, we're going to tend to be at the maximum. And we've seen a plot at the bottom right, kind of how the QALY benefit changes within each one of these bounds, the preference share, the peak preference share. We assume that preference share is going to rise to this peak over 5 years, so 5-year uptake, and then we're going to have 10 years of market exclusivity with rapid loss of sales after LOE. Now later, we're going to adjust all these assumptions as we proceed to account for inflation effects due to the longer survival of treated patients. For other commercial assumptions, again, we're trying to put in ballpark estimates that are kind of illustrative of each class of disease. For list price, ultra-orphan is quite expensive at $350,000, orphan at $150,000, specialty at $50,000, primary at $10,000 per patient year list price. We apply a gross margin that gives us a COGS that's going to be more expensive for ultra-orphan, much less for primary, and we have that price then grow modestly over time. In the US, for access, we assume that payers impose a prior authorization to label, but within the label, patients have pretty strong formulary access, and so only see a 20% haircut for things like out-of-pocket expense. Gross to net rate in the US, we vary based on the degree of competition, and so we have a very low gross to net rate in ultra-orphan and orphan, in specialty and primary, where we have head-to-head competitor with one other drug, we have to pay a lot more in discounts and rebates. In the EU, as I said, by default, we're going to assume that if we are reimbursed, patients have 100% access, no out-of-pocket expenses. But what's key is the gross to net rate. So we have, by default, discounting is required to hit that cost effectiveness target. But since that may be less than COGS, since the cost of goods sold, we also model two alternative options, one in which the manufacturer decides not to launch in the EU, and the second is where EU payers will pay for the drug through a special provisioning scheme at the list price, so not by applying cost effectiveness. Now, one nuance of the forecast here is that, as we see, when patients are treated, it slows down disease progression, and so they survive for longer. And the net result is an inflation in prevalence, in the fraction of patients who pass inclusion criteria, and in the peak preference share that, as we see below in a second, can be quite significant when the treatment effect is large. I won't walk into all the details of the derivation, but we show the basic results here. The idea is, if you take a quantity like prevalence, before the effect of inflation, it's quite a simple formula. It's the incidence times the mean time scale of progression among patients. After inflation, you have to account for the fact that patients who are passing inclusion criteria, put on drug, and have persistence end up surviving for longer, and that makes that population larger. We have similar assumptions for the other values to compute peak multipliers for prevalence, the fraction passing inclusion criteria, and peak preference share needed in order to describe the inflation effect. And these multipliers grow to these peaks over a time scale that's the longer of uptake or the turnover of the population that passes inclusion criteria. If we look at the net effects for linear ultra-orphan at the bottom, we see in each one of these areas an increase in prevalence, increase in the fraction passing inclusion criteria, increase in peak preference share, and it yields in total about a 57% increase in sales due to all these inflationary effects. And so that's quite significant. It's a smaller value if we're looking at primary where the treatment effect is smaller, but it is something that we definitely need to consider when we forecast the economic value of these drugs. Once we include inflation effects, the rest of the model is pretty much a standard type of prevalence model. We take the total prevalent population, we apply the fraction passing inclusion criteria, apply the access, uptake, and inflation adjusted preference share, and compute the number of patients that are active on drug at any one period in time. We then apply assumptions around the dosing. Here we're assuming QD dosing, so once a day, adherence to get the number of drug units consumed over a period of time, multiply that by the list price to get gross revenues, and then apply a gross to net rate in order to get the net revenues. Here for the linear ultra-orphan disease, we see the sales, costs, and earnings before interest and taxes when we have the case that all development succeeds. For patients, drug units, gross revenues, and net revenues, we break out these quantities by US in blue and EU in orange. We see that because of this very strong discounting in the EU for cost effectiveness here, we have very little net revenues that come from the EU, even though there's considerable volume. For the costs, we have the development costs of each trial and regulatory review. We have assumed commercial costs that we have to incur every year to support the product, and then we have the cost of goods sold, again, separately driven by volume in the EU and in the US. Now, each one of the revenues and costs that we include in the model are tied to the development program outcomes. We only incur the cost of a clinical trial if we execute it. We only see net revenues if we get regulatory approval. What we do then is we calculate expected net present value by considering all the different possible outcomes of the development program using our clinical trial simulations to compute what the probabilities of success are at each one of the stages. So here we show in the pie chart the distribution of outcomes labeled by the highest state that's achieved successfully. The last one is full approval. This is where we're on the market for the full time. That occurs in about 82% of outcomes. We see the other failures at intermediate stages along the way as the other elements of the pie chart. In each one of these outcomes, we separately value the drug program, and that's what we see at right with the net revenues, costs, and EBIT for each one of the different possible development states, starting from failure at P3 at the top, where the only thing that's successful is that we start phase 3, where we have no revenues, no sales. We have the costs of the single phase 3 trial. P3 success, we're successful in phase 3, we move on to regulatory review, but we fail that. Again, no sales. Now we have slightly extra costs. In conditional approval and P4 success, we do initially get sales because we've been initially approved, but then either the phase 4 trial fails or its regulatory review fails, and so we're then pulled from market after only a few years. We incur costs during the period we're on market. We get a slight period where EBIT is positive, but it's still highly limited in terms of economic value. And finally, at the bottom, in full approval, we have the economics that we just saw if all the different steps of the trials are a success, and we have sales as long as we maintain market exclusivity for 10 years. We take the values of the program for each one of those individual outcomes and then average those, weighted according to the probability of observing each outcome, to get expected net present value, shown at left on a pre-tax basis and at right on an after-tax basis. In addition to the tax rate for the orphan designation, we assume a larger R&D tax credit as well. What we find here for linear ultra-orphan is the after-tax expected net present value is relatively low at $177 million. Again, we're starting already after a successful phase 2, so the expected value is going to be a lot higher than it is if we consider earlier stages of clinical development. And again, we're assuming here this base case of early mid-stage inclusion criteria and a 1-year protocol duration for the trial. We see that the cost of the failures are relatively cheap because we have small single-arm trials, and even a very short period of sales with the conditional approval is enough to at least break even on that. We also see that despite the very high preference share, strong access assumptions we're making, the treatment effect inflation, which is very large, all of these are very optimistic assumptions for such a disease. The value with full approval is still limited by 2 factors, the small patient population, and as we see in the EU, the low pricing that would come about if cost effectiveness framework is applied. Here we see a broader set of economic metrics for the ultra-orphan late-stage program, both linear, which is what we've been considering to now, and non-linear, which I'll get to in a moment, which has some interesting differences. First, on the linear disease, in the center column, we see that we can use relatively small trials because we have such a strong treatment effect, but even so, we have to go to nearly 1 in 5 of the available centers of excellence in order to recruit enough patients to close it. Again, showing ultra-orphan drug trials are extremely difficult to close. It's one of the main reasons why, of course, we use these synthetic control single-arm trials. Development costs are low because we do have small patient counts, so the economics of developing it are pretty cheap. But what we see in linear disease is that the cost-effective target price that would be calculated in the EU is actually lower than the cost of goods sold because we're assuming this is a relatively expensive drug to make. As a result, in the EU, if we decide not to launch in the EU, we actually see a higher expected net present value than we do in a base case where we launch, but have to discount it steeply because we have all those COGS that we're accruing and potentially also additional commercial costs. That being said, if we did want to launch it in the EU and we did it in a bare bones way, you could still do it without paying too much economic penalty. But essentially, you're not getting any economic value out of the EU. It's all coming from the US if you have, rigorous cost effectiveness pricing that's applied. Now, the nonlinear disease is different in interesting ways that have some immediate economic implications. The 2 main ways that we see across the board nonlinear disease is different is first, there's a greater fraction of patients that pass the inclusion criteria when you have restriction to this early and mid stage of disease. And that's because in linear disease, patients quickly proceed to that late stage so quickly, they just remain longer in those earlier periods of disease. Second is that because patients do hang out with higher initial quality of life for a longer period of time, you can initiate therapy before you have a lot of that later irreversible loss of quality of life. And so from a cost effectiveness standpoint, your qualities go up because you're effectively able to intervene earlier before you get as much reduction, impairment in quality of life from the disease. And as a result, for nonlinear now, the target cost effectiveness price is actually higher than COGS, still not incredibly high, but it's economically favorable to launch in the EU. And overall, you're making more revenues also in the US because more patients pass the inclusion criteria. That being said, we're still talking only about a few $100 million in terms of peak net revenues. Expected net present value is relatively low. And so ultimately, you do find that the commercial value here can be strongly limited by just the number of patients that are available. If we go to an orphan disease, here not a very large one, again we're assuming only 20,000 patients in the US, we find a similar set of economic considerations, but ultimately the economics are a lot more favorable than an ultra-orphan. We have to use some bigger trials because the treatment effect is not quite so large. But now, because there's 20 times more patients, we don't need to go to so many sites in order to recruit them. The development costs are still relatively low because we're not talking about very large trials, even though now we have 2 arms. They're not incredibly expensive. And we find that the economic potential now gives you the possibility to have blockbuster sales above $1 billion. Now here we're still assuming something that's relatively optimistic in a sense that there's no competition in this orphan disease, and among the diagnosed population we're able to get pretty broad awareness, activation starting on drug. We know that in orphan diseases, particularly if there's never been a indicated treatment for that disease before, it can take a long time. It can be a challenge to get patients kind of activated on drug. We're not assuming any of those haircuts yet in here, but we see much higher potential value in this disease compared to what we saw in ultra-orphan. When we move to specialty, now we have an order of magnitude again, more patients that are available. And so because of that, we're going to assume a traditional 2-trial regulatory pathway. And we're also going to now assume that we need to have more patients in each one of these trials in order to appropriately measure safety before we get that full approval. And so here we're using much larger arm quotas, again, that are actually overpowered from strictly an efficacy power standpoint, but is something that may be required by regulators in order to get more patient exposure to drug so that we can detect less common side effects. The trials now, because they are larger, are significantly more costly. We also now look at not just a 1-year protocol duration, but also a 3-year protocol duration, which of course further increases the length of time of the trial and significantly increases the cost of the trial as well. We see again in the cost effectiveness price that we don't get a very large target cost effectiveness price in Europe, but at least it's again above the COGS, and so it's economical to launch in Europe. And also because the list price itself is coming down, the actual gap between the US and the EU is still significant, 3 to 4 times the price in the US, but at least now the gap is closing a little bit. In the peak net revenues, we see that we can get well above, in the blockbuster area, 2 to 3 billion, potentially more with nonlinear that we're talking about in a second. Again, we're assuming here that we have only a single head-to-head competitor, and so that's a relatively optimistic situation because now we're getting into a larger disease. In nonlinear disease, we find the same kind of advantages that we saw before, where more patients pass inclusion criteria when we restrict it to early and mid phase, and we see greater credit in quality adjusted life years because we're effectively able to initiate treatment earlier. And so the effect of both of those is to give us significantly higher peak net revenues and expected net present value for the nonlinear disease relative to the linear disease, with otherwise similar types of assumptions commercially. In specialty, the base case that we just showed effectively had one head-to-head competitor. So we said peak share was somewhere between 30 to 60%, depending on the quality benefit, and the US gross to net rate was 50% because we have to discount significantly or give rebates in order to get formulary access. What we do here is we consider 3 additional competitive scenarios, one in which we have no competitors, one in which we have 2 head-to-head, and one in which we have 3 head-to-head. And so those peak share declines, and then in the US also as well, we see a change in the gross to net rate. Again, all this is very ballpark, but it's saying that we get in a competitive market, we have to fight harder in order to get peak share. We get a smaller amount of it, and we have to give away more value to payers in order to be the preferred agent on formulary. What we find in both linear and nonlinear disease is as we have a more competitive market, we strongly degrade the peak net revenues and the expected net present value. So much so that once we get to the situation of 2 to 3 competitors in these diseases, we're actually don't appear to be creating that much more value than we did if we had an orphan disease that was non-competitive. So again, specialty, more patients, bigger potential, size of the prize if we get it all to ourselves. But again, what you typically find is that oftentimes that very size of the prize pulls in a greater number of head-to-head entrants that may have similar mechanism of action, similar type of modality, unless you have very differentiating IP, and it can end up impairing the overall program value compared to what you would see if instead you pursued something that was maybe a smaller disease but less competitive. If we move to primary disease, we see very similar types of dynamics. Now, we always look at the 3-year protocol duration because we have such a longer time scale in here. And what we find economically is we do have 5 times more patients, but the list price is also 5 times lower relative to specialty. So we see kind of similar ranges for things like net revenues. We do assume that there's going to be more commercial costs, and so generally speaking, the expected net present value is a little bit lower. We also, particularly in nonlinear disease, have to go to higher trial sizes, so development expenses are also higher. So if we go to nonlinear disease, it's important to kind of understand the drivers on why that behaves differently than linear disease. Once we get into the regime here where we're actually limited in power, so we're designing trials on a base case to have 85% power in each trial. That gives us about a 65% overall probability of technical and regulatory success when we consider the 2 trials, plus the need to have no safety signal at 10% and have successful regulatory review. But what we find is in nonlinear disease, yes, we still have advantages of more patients pass the inclusion criteria. We still see an improved benefit under cost effectiveness because we effectively start treatment earlier. But when we have a more variable patient population in the trial of not just timescales varying across individual patients, but also across individual stages of those patients, whether they're early versus mid-stage disease, we find that we need a significantly larger trial in order to hit 85% power in nonlinear disease than we do in linear disease. As a result of that, here now we introduce these 2 additional inclusion criteria, early, mid, fast. So we just pick the patients whose prior treatment history suggests that they're on the faster end of progressors, and mid-stage disease. And we find in each one of those we can, in fact, achieve 85% power with smaller trials that are less expensive. The problem is if we look at the number of patients that pass inclusion criteria, we drop that from 92% before inflation to 34 to 38% when we use these more restrictive criteria. So we have the potential, if that's reflected, for example, only fast progressors would be included in the indicated use of the label. Payers would restrict reimbursement to those patients. We would be giving up a significant amount of revenues because of that and see then a much lower potential on peak net revenues. With the mid-stage patients, we get the same effect, but it's even worse because now we're starting therapy much later. So in the EU, we make out significantly worse under the cost effectiveness assessment. And so our revenues now go down in Europe because we are essentially not being credited as much under health technology assessment for the value that we bring because we can't recover any of that quality of life that was lost before treatment began. If we look at the effect of competition on economics in the primary disease, we see the same kind of trends as we saw in specialty. As we move from the base case with 1 head-to-head to have 2 or 3 head-to-head players, we have lower peak share. In the US, we have to give up more on gross to net through rebates and discounts. And so the peak net revenues and the after-tax expected net present value decline, again to a point where even though these are much bigger markets, we're effectively not creating more economic value than we would if we had a smaller orphan disease that was non-competitive. So again, high size of the prize, oftentimes without strong IP, pulls in a lot of individual entrants, and it ends up creating a challenging market for everyone, as opposed to these narrow, smaller diseases where maybe you have a more differentiated IP that allows you to realize a significantly more amount of value, even if the patient population is smaller. Now, until now, we've been using these economic calculations in order to select the optimal program design among qualitative factors. So should I use a 1-year protocol or 3-year protocol? Should I use a broad, early mid inclusion criteria, or should I try restricting it in order to enable a smaller trial? But you can also use this approach in order to optimize things like the size of the trial, so the arm quota, and see how do I change those parameters that can have many different values, and see what is the design that optimizes the economics here, expected net present value, at least within the range that we assume is going to be needed on clinical or regulatory reasons. Here, what we do for nonlinear primary, we saw that on a base case with a target of 85% power with 760 patients per arm, we had about a 65% probability of technical and regulatory success. What we want to do is we want to vary the number of patients per arm to the size of our trial and see how it affects the probability of technical and regulatory success, development cost, and expected net present value. So we do that by changing the arm quota, and for each one, rerunning many different clinical simulations of the trials to calculate duration, cost, success probability of each stage, linking that to the overall commercial forecast to calculate expected net present value. What we find at left, as we expect, is that as we go to larger trials, the power increases and the probability of technical and regulatory success goes up to this upper limit, approaching about 85%, where you have failure primarily due to safety and at the regulatory stage, but you've really kind of done all you can to maximize the power of the trial. Development cost, as you would expect, keeps on increasing as we go to more and more patients. And expected net present value reflects the trade-off of these. If I go to increased investment, I see higher probability technical and regulatory success. That gives me a better chance of realizing all that value from sales of an approved product, but it also increases development cost. And once I get up into a region where I've gotten diminishing returns, I can't do that much more to increase the probability of technical and regulatory success, then you start to see expected net present value go down. Here we have an extremely high opportunity in terms of the net present value. When we have a product that's successful, it's billions of dollars because it's a huge disease. And so this is telling us that we would want to make extra investments to have something like about 1,400 patients per arm to really maximize the power on it and be able to achieve the highest possible chance of success. Here, I should say, as we increase the number of patients per arm, we're also increasing proportionally the number of sites in our network, so we can continue to recruit the trial in about the same time period. As we go to smaller diseases like ultra-orphan, if we can't really go very much broader on the number of sites in the network, what we find is we go to too larger trials, it just hangs on forever, and so we start to see an earlier decline in expected net present value because we've exhausted the patients and just the trials take forever to close. And before we found the optimal value just by changing the different clinical design values, and then for each one doing all the program simulations, averaging the top line data that we see when it's successful, calculating the resulting forecast. There's a lot of effort associated with generating each one of those points, but there's a structure in how the clinical and commercial factors are coupled together that allows us to more effectively see how that optimal design is influenced by the commercial assumptions we make and the uncertainty in those assumptions. And so I am going to walk through this even though it's a little mathematically here. But the basic problem we want to do is we have some set of design parameters, theta, that we want to vary to maximize economics, maximize, in this case, expected net present value, within some bounds of a region that's going to be dictated by what regulators expect, what we think is, clinically supportable and operationally feasible. Now, within that region of possible designs, we want to sample at random a couple of different designs, different size of trials, different durations, whatever we decide to change. And we do our program simulations to calculate the probability of technical and regulatory success, the present cost of development, which is all the development costs discounted back to the evaluation date according to the cost of capital and summed together, and the launch date, and any of the top-line metrics, such as the qualities that we would need to calculate other assumptions in our forecast. We calculate values of those from these clinical program simulations for a different sampled value of the parameters, and then we just fit those values to some interpolating function that we can use to see more broadly as we navigate around in this space, what's the impact on development costs, on launch date, on top-line metrics, on probability, technical, and regulatory success. And once we have that from the commercial forecast model, we can compute the present value when development succeeds of the net revenues less COGS and commercial costs that are only interact with the clinical simulations through the launch date and these top-line metrics, and then they depend on all the other commercial assumptions that we put into phi here. So we can calculate that easily. We just need to know the launch date, we need to know these top-line metrics, we need to know the other commercial assumptions, but to calculate that, it's just a run of the forecast model. We don't need to go through all this program simulation. If we put those things together for a set of commercial assumptions, we can calculate the economic optimal program design by then just slotting into our forecast model the launch date and the top-line metrics that we estimate from interpolating between these clinical program simulations. The other parameters we use, the probability, technical, and regulatory success, the present costs of development, are ones, again, that we're interpolating from the few trial simulations. The nice thing is, once we do a set of those trial simulations to get that mapping, we can change all the commercial assumptions that we want and see how that influences the resulting optimal design. As we have higher value of net approved products, we expect to support greater investments, bigger trials than if we have a low value drug that, even if successful, is only going to yield a couple $100 million in sales. You're not going to put the same investment into that trial design because that present value of success is going to be lower. Now, with commercial uncertainty, where we don't know exactly all the inputs, but we can estimate and draw random values, between lower and upper bounds, what we can do is calculate an average present value if development succeeds by averaging over the forecast values in our model for different sampled values of the launch date and top-line metrics. And we can use those from values that we generated in a clinical simulation earlier. Again, that's relatively quick because all we're doing is running the forecast model. Once we have that sampled values of this average, present value, we can again fit an interpolating function against those and then slot in the actual relationship of launch date and program metrics with trial design, and now get a formula that we can use, again, relatively quickly to calculate what's the optimal program design averaged over all the uncertainty that we have in the commercial market. And so what this approach allows us to do is to take kind of the elements that we've seen before and have a very rigorous combined clinical and commercial evaluation of the program to see what do you have to believe about the market for this design to work. How does that change if we believe different things about the size of the market, our ability to gain share, and our ability to get access at a favorable reimbursed price. So to summarize, we just show in one plot here kind of what the late stage economics look like for our base simulations And we do this before we've done any optimization on program size for simplicity, but for any one of these diseases, obviously we could use the approach that we just discussed to navigate around alternative program designs. For each one of these simulations, we have a label that is going to be the prototype, ultra-orphan, orphan, specialty, primary, the shape, linear, nonlinear, the EU reimbursement assumptions, either reimbursed with cost effectiveness or reimbursed at a list price, only if we have, an ultra-orphan or orphan drug where cost effectiveness, doesn't come close to working. And then the number of competitors. Base case is 1, and then we have 0, 2, or 3 as less or more competitive cases. So we see on x-axis the development cost, so that's the amount of capital that we're putting at risk in order to launch the late-stage program, and then on the y-axis the expected net present value that we see. Here I don't report it in terms of return on invested capital because the numbers are incredibly high compared to R&D overall, because of course we're starting with a late-stage program with established proof of concept, dosing and intervention model that we're going to keep constant into phase 3. And so again, we're starting already from a very, very favorable spot, and it's a question about how do we execute this last stage in order to get it over the finish line. What we do see is some interesting trends and insights, I think, that we should keep in mind if we're looking at portfolio design, which of course oftentimes we're limited to what we have in the pipeline, right? But from a acquisition standpoint, from deciding kind of R&D priorities, again, there are some insights in here on managing portfolios that I think are useful to keep in mind. The first thing is that we see, again, competition is so incredibly important to expected value in specialty and primary. Large diseases, large size of the prize, they have a tendency to attract a lot of entrants that are doing similar things, all coming to market about at the same time. That can really impair the value to any one of those entrants of a program that's launched in that disease. By far the best thing is to have a big specialty primary disease and to have it to yourself because you're printing money, right? That's where you get the expected net present value above $10 billion. Mega blockbusters, huge successes. There's only a handful of those in the industry. If you look at ultra-orphan and orphan and compare those to specialty and primary, ultra-orphan fundamentally is limited by the smaller number of patient counts, and again, potentially also in the EU by the cost-effectiveness treatment. And so it's really hard to get a super blockbuster drug out of an ultra-orphan disease. But orphan drugs can look much better because there's still relatively few patients. Some orphan diseases, certainly many of them, are somewhat competitive, but you have a better chance of maybe having at most one major competitor in some of these diseases. Again, order of magnitude more patients than an ultra-orphan, but the price is only half or a third, and so you do have a significant revenue potential. And you can see similar kind of expected net present value from one of these uncompetitive orphan programs as you do from a highly competitive specialty or primary. But importantly, you can get that expected net present value with less capital at risk because the development costs are lower. They're lower because the trials are smaller, and if you have a conditional approval pathway and you fail on safety, you only have to spend money to do one trial to find that out and not two at the same time, and they're both a failure. And so for the same amount of capital, to do one large competitive late-stage program in specialty or primary, you can maybe do 2 or 3 in orphan, and not just have a higher expected net present value overall because now you have 2 to 3 programs instead of 1, but that can also be de-risked unless you're using a common platform underneath those. And so there is a pathway to have an attractive return on late-stage invested capital in these orphan programs that in some ways is a little bit of a sweet spot that's consistent with the thesis published and pursued and published recently by BridgeBio, as outlined in the paper cited here in Portfolio Management Research on some of the diversification and economic advantages that you have by going to some of these smaller, maybe less competitive diseases, as long as you can get a big treatment effect because you're treating the underlying driver of the disease. So conclude with just a few kind of takeaways from the study that we've done here. The first is, by taking the available data that you have, phase 2 patient registry data, and using clinical trial simulations in order to get things like program duration, cost, and risk, you're reducing your dependence upon finding the right set of analogs for those estimates that you think are going to most translate to your program. And that can be somewhat subjective in how you do that. The second is, even though we've used very rough and simplistic estimates of cost structures, commercial dynamics, and kind of doing it for these very, very broad disease prototypes that have a lot of intra-prototype variation, we do find that the resulting economics reflect what we see in real life. Some of the challenges that we see in a recruitment standpoint, an economic standpoint in ultra-orphan, and the impact of competition in much more larger diseases. We also, through this approach, can highlight some of the more nuanced aspects of the disease dynamics that end up having significant economic impacts. The inflation due to treatment effect, as we saw, a 57% rise to sales in ultra-orphan. The influence through quality-based access and target pricing, potentially in the EU, and how that links to the shape of the disease and nonlinear disease, where you have this accelerating function decline in late-stage disease. There's a lot of ways that the economics look different due to the underlying dynamics of the disease, which may not be immediately apparent unless you do this kind of integrated clinical and commercial simulation. Second is, what we've done here is really kind of map detailed program designs to program economics in order to be able to see what is really driving the value. What are the changes that we can make in terms of how we design our program, and how does that allow us to see how do we steer the program to more promising economics, as long as we're within bounds, again, that is going to be set by operational feasibility, clinical concerns, and regulatory restrictions. And again here, what we've done it for are toy diseases. We've tried to make them somewhat realistic, but the key thing is the same approach could be applied to programs in real life using inputs that are largely developed today, right? A lot of the kind of things we're doing in clinical trial simulation, clinical teams may do in order to calculate the size of the trial or to investigate alternative designs. Certainly from a forecasting and valuation perspective, a lot of the economic analysis is done, but uses analogs for things like program duration, cost, and risk. The key point here is to integrate a lot of the work that's done today across the organization into a unified view, and to see how those factors interact to end up influencing the optimal late-stage design and set of decisions. So with that, I'd like to conclude, and thank you for your time.